Welcome back, Year 12s. For this video, we need to be aware of the lymphatic system. So this is where your lymphocytes travel, specifically those antigen-presenting cells we've been aware of. Antigen presentation, which is thanks to our, of course, APCs, such as dendritic cells, all right, B cells, and macrophages. And they're going to be finding a complementary T helper cell to be able to activate them. All right, it looks something like this. All right, you get your B cell, you get your T helper cell, as long as it is the proper match. All right, the MHC2 markers will display the antigens of the foreign pathogen, and they'll get activated, which means they can do their job. We're looking at the humoral immune response today, and again, remember this happens concurrently with our cell-mediated immune response too. So humoral immunity, what is it? It involves B lymphocytes specifically, all right? And they're going to mature in that bone marrow there, okay? They produce specific antibodies. So these Y-shaped proteins are the antibodies, all right? And they really do all of the work, all right? The B lymphocytes just differentiate and be able to produce them. They're released into the blood and the lymph, and then they'll eventually find the pathogens and destroy them, all right? There's other macromolecules that are involved too, such as the complement protein. T helper lymphocytes are also involved. They need to be the ones to activate the B lymphocytes. Obviously, that's going to happen in the lymph nodes, all right, which is where these two cells will find each other. All right. So we have T lymphocytes here, okay? So these T helper cells, all right, they allow the APC here to be activated. So we have the MHC2 marker, which is displaying the foreign antigen, okay? And once that finds its corresponding match, then our T helper cells will shoot out cytokines and will activate our APC. In this case, it's going to be the B cell, because we're looking at the humoral immune response, all right, which will allow it to go through clonal expansion firstly, which means it duplicates itself, goes through mitosis, and then they differentiate, okay? So our B cell here, our B lymphocyte, will differentiate into both B memory cells and plasma cells, whereas our T helper cell will then differentiate into T memory cells. Again, these plasma B cells are super important. This is what we're looking for here, okay? So they're derived from the activated B lymphocytes. So once the B lymphocytes are activated by the T helper cells, once they've found the corresponding T helper cell, they essentially clone themselves through clonal expansion and then differentiate, okay? And the plasma cell is one of the two differentiations, all right? And what they do is they do the job, okay? This is really what we're looking for. Plasma cells start to create the antibodies, all right? So boom, boom, boom. The antibody looks like this shaped protein here, okay? And they have a bunch of different roles in destroying pathogens. All right. The other differentiation of the B cell is the memory B cell. So derived from activated B lymphocytes, one of the two differentiations lingers in the bloodstream for up to 100 years, and it remembers that particular pathogen, okay, and the antigens found in them. So really, that's what we call the immunological memory, all right? And that is super important. All right, so the example we commonly give is, survivors of the Spanish flu have had their blood tested, and they've seen that 50, 60, 70 years later that they still have these memory B cells in their body, and therefore they still have immunity to that disease. All right, thanks for watching, guys. Have a good night.